afternoon, my name is Aimee Fowkes and I'm both a PhD student at Newcastle University, but my day job is at Farah Science Limited. And the work I'll be talking about today is actually from my work at Farah, not my PhD. So Farah is a diagnostic lab. We're based in York, and we are the National Reference Laboratory for Plant Health in England and Wales. And we do both commercial and statutory testing as well as research. Our statutory testing involves inspectors around the country, collecting plant samples. So that could be at airports, any plant samples coming through, they will inspect and might come to Farah for testing. So we're interested in tree viruses because the most recent publication on them was from 1980. The same latest publication, the most comprehensive, covering a number of tree species. Since then, there has been some odd reports of viruses in trees, that we'll talk about a little bit. It's really important for us to understand what viruses are present in the UK, baseline, you know what's there, what could be a threat to our UK trees. And today I'm going to talk about some work we've done surveying for them, using both leaf material and pollen. So for this work, we had help from the observatory volunteers. Observatory is a citizen scientist group who are trained to look for potential threats to UK trees, such as these ones. We have all kinds of pests and diseases, including one virus, just there. That disease is mountain ash ring spot, caused by European mountain ash ring spot associated virus. And this virus infects sorbus, mountain ash, and it's widespread across the UK. It causes these ring spots on trees. It's very obvious to see. Once you've seen it once, you'll see it everywhere. More recently, for our work on this project looking at viruses in trees, through a volunteer, we've also reported this virus on chokeberry and juneberry for the first time in the UK. So you can see this work is really helping us understand not just the distribution of the virus, but its host range. And if you are about and you see some ring spots on trees, you can report them to Tree Alert through Forest Research. Again, that can help us understand this viral distribution. So, the particular virus I'm going to talk about, we first found at the Yorkshire Arboretum. This arboretum is based about 10 miles from where I work, and occasionally, a couple of years, as a team we go out to the field to look at virus symptoms, look at trees, because, as I said, we get all our samples by inspectors. So we get really good at looking at samples in bags, and not so good at looking at the tree as a whole thing. So going to the Arboretum gives us a really good training experience and understanding of how these things may look in context. So while we're out there, we came across a tilia tree, which you may know as lime, linden or basswood. And this particular tree had some symptoms we'd associate with infection of virus. These are smaller leaves, twisting of leaves, and this banal chlorosis. And through some testing, we found a novel virus, which we named Tilia nicovirus 1. Nicovirus is the taxonomic placement, that's why that word is in there. So, okay, we found this virus, great. But what does this mean for UK trees? Is it causing symptoms? How widespread is it through the Arboretum? To start answering those questions, we had a summer student, Aimee Williams, and she collected more tilia samples from across the Arboretum, shown in pink here. And of those 53 samples, 16 of them were positive for our Tilia nicovirus 1. Importantly, this virus was present across a range of Tilia species and varieties, so it's not restricted to one tree or one species, and there was no association with symptoms. So, as of yet, we don't really understand what this virus is doing in trees or if it's having an effect on them. So, we've got a distribution. We know it's widespread in the Yorkshire Arboretum. What about the rest of the UK? And this is where our observatory volunteers come in, because they're present across the country. And through this project from the Bayou Fresco, the Spinotry project, they collected all manner of trees, oaks, pines, ash, sambucus, everything you can think of. I'm only talking about Tilia today to keep it simple. But over the last 2 years, they've collected 81 samples, as represented by these blue trees. Very roughly, not the right numbers, but you get the idea. Of these 80 trees, 28 are positive. And what this has shown us, the trees in pink, the positives, purple, is that this virus is not restricted to the Yorkshire Arboretum or Yorkshire. We've got a finding in Wales, East Midlands, Cumbria, Northumbria, and up into Scotland. And not yet the south. But we could do further testing and see if it's also present there. So, everything I've told you about so far is based on leaf material. So there's people going out, testing trees, finding trees, sampling them, sending us the material in. But we were able to look at this survey for this virus in a slightly different way. So through our testing, we were able to get a DNA sequence of our virus, and we compared it to an online database. And through this, we got a match to a previously published sequence. This sequence came from a paper looking at bee-infecting viruses in Scotland. Now, our virus is a plant virus, we didn't think it was in bees. What we thought was that actually the virus in the tree got into the pollen, and when the bee's been going around collecting pollen, it's picked it up alongside, and that's where the sequence has come from. And because it's found in Scotland, we got in contact with our Scottish counterparts. So, Parasites Limited is the National Reference Laboratory for England and Wales. SASA is the same for Scotland. And they have a bee health unit monitoring pollen baskets, and they went back into their freezer, pulled out some samples and tested them for Tilia nicovirus 1, and we got 3 positives, showing that the virus is present in pollen, and also we've got more findings in Scotland. So, we know the virus is in pollen, and maybe we don't want to walk around and collect a bunch of trees, leaves and trees, because Tilia are a really common amenity plant. So if you go into cities, they're often lining the high streets, there's hundreds of them. What if we can make bees do the hard work for us? Because they're already doing it anyway. So bees have about a 5 to 8 kilometre flight radius from their hive. They'll collect pollen in their pollen baskets. And if they're out and about collecting pollen, if we test the hive sample, and again we're reducing the number of samples we have to test, if we find positive there, that suggests in the wider environment, in a radius around their hive, there could be a positive, which could help focus our surveillance efforts. So last year, we had a summer student called Jamie Hewitson who investigated this work. So they got in contact with Yorkshire and Beverley beekeepers, and they were sent samples from 28 hives, some of which are shown here, and 10 of them tested positive. So if it's a light blue bee, it was negative, and yellow bees are positive. As well as testing the hive, again, we can't replace leaf material testing entirely, so we also tested some trees around sites of the hives or positive hives. Again, blue is negative and pink is positive. And we can see that there might be, where we're finding these positive hives, suggests that there could be, we have been finding these positive trees in the same area. Again, back in this idea that we could use these to focus our surveillance efforts. So, to conclude this work, we initially identified this novel virus in Tilia. We have shown that it's not associated with symptoms. Through our sampling of trees across the UK, we've shown that this virus is present in Wales, across North England and Scotland. And of the 171 samples that have been tested as part of this work, 58 were positive. And then we've also shown that this virus is present in the pollen, it's both pollen baskets and beehives, so that was the pollen and hive debris of positive virus. giving us this alternative way to survey for plant viruses. And my last thing to do is to thank everyone involved. So that's the virology sequencing teams at FERA, both past and present, our summer students. So we have Aimee Williams, who is now a member of my team, Jane Hewitson, who got last year, our colleagues at SASA, Forest Research and the Yorkshire Region, our funding bodies, and last of all, just to thank the volunteers from Observatory and Yorkshire beekeepers and Beverley beekeepers, without whom none of this work would be possible. And thank you so much for listening.